check, mic check. Alright. Hi everyone.
Hey.
Okay, so as you can see, we're now in the middle of somewhere to discuss something related for our podcast today. So without further ado, let us invite the first speaker, Esther, to begin the session.
Hey everyone, welcome back to Science Seeds, where we talk all things agri-tech and sustainability. Today's title is Applying Omics Technology for Crop Improvement, a functional genomics and proteomic approach for sustainable agriculture and climate resilience. We are from Group 2, Subgroup 4. I'm Esther, and joining me today are Alexis, Elizabeth, and Angel. So today we're diving into how functional genomics and proteomics are changing the way we approach agriculture. Now here's the deal. The world population is heading towards 9 billion by 2050 and it's expected to peak at around 9.73 billion by 2064. That's a lot of mouth to feed. But agriculture is under pressure. We're facing a shortage of land suitable for cultivation. There's less freshwater and climate change is making things worse. More drought, more floods, and unpredictable weather.
Right, and on top of that, we've been relying too heavily on agrochemicals and antimicrobials for decades. While they've helped in the short term, they're starting to backfire, lowering the long-term productivity of traditional farming. So yeah, food security is at risk, especially in places where crop yields are already plateauing or falling. That's where science steps in, with tools like functional genomics and proteomics offering smarter solutions.
Exactly. So let's break it down. Functional genomics looks at how the genome and its products, like RNA and proteins, work together to control plant growth, development, and stress responses. And then there's proteomics, which focuses on the actual proteins, the end products of gene expression. It studies their structure, function, and how they respond under different conditions. Together, they help scientists identify the key traits in plants that influence yield, stress tolerance, nutrient uptake, and disease resistance.
And here's where it gets interesting. Recent study shows that combining genomics and proteomics, and even other omics like transcriptomics, can give a much clearer picture of how plants react to stress. Researchers have already found major genes and proteins linked to drought, heat, and disease resistance in crops like rice, maize, and even durian. In Africa, there's a real need for this technology because of climate and pest challenges. But sadly, lack of infrastructure is holding back the progress. Still, proteomics and functional genomics are already making a big change. They're helping to develop stronger, healthier, and more sustainable crops, which means we've got better tools to fight hunger and to protect food security.
Now that we cover the basic, let's talk about how genomic is being used in real crop. This part all about result, what actually happening on the ground with genomic based tool.
Absolutely. With climate change, resource depletion, and the rising global food demand, crop improvement has become more important than ever. In fact, if we don't do something, global crop yields could drop by 10 to 25% by 2050, especially in tropical and subtropical regions.
That's where genomics plays a key role. It helps us understand the genetic basis of traits like high yield, stress resistance, and nutritional content. Techniques like MAS and GS let breeders work faster and more precisely to improve crops.
Let's start with field traits. In rice, genomics has helped to identify genes that control spikelet development and photosynthesis efficiency, both of which improve grain production. Meanwhile, in maize and sorghum, genes have been mapped for better nitrogen use and water efficiency, which are the key traits to survive dry spells.
And when it's come to climate stress, Genomics is super useful. For drought, scientists use RNA sequencing and genomic-wide SNP analysis to identify genes like aquaporin, late embryogenic abundant protein, and those involved in ascorbic acid signaling. This help rice plants hold onto water when it's scarce.
Additionally, for heat stress, wheat and other cereals like barley benefit from the discovery of genes that trigger heat shock proteins like HSP70 and HSP90. This helps stabilize proteins and membranes when temperatures rise. Imagine editing crops to survive heat wave. It's already in progress.
And then there's salinity. Using QTL mapping, researchers found key genes like sodium ion and hydrogen ion antiporters and RS scavengers in salt-tolerant rice. This is a huge deal for coastal areas with rising soil salinity.
But it's not just about stress. Nutrition matters too. In Musangking durian, scientists use GWAS technology to identify genes involved in sulfur metabolism, sugar transport, and ripening control. And for biofortified rice, genes controlling iron and zinc accumulation have been mapped to help fight hidden hunger.
Okay, so those are powerful examples. But what about fighting pests and diseases?
Genomics has given us tools to do just that. Through sequencing and GWAS, researchers can identify resistant genes and even use CRISPR-Cas9 to enhance plant immunity. For example, the CSLOB1 gene in grapefruit was edited to improve resistance to citrus canker.
Plus, in sorghum, GWAS revealed that genes like ERF113 help the plant resist striga, a destructive parasitic weed. These genes are tied to cell wall strengthening and defense signaling.
And let's not forget about maize. GWAS has also helped to identify the genomic regions, or QTLs, that provide resistance against multiple diseases, Fusarium earrot, grey leaf spot, and northern corn leaf blight. This helped breeders to develop disease-resistant hybrids without needing decades of trials and errors.
QTL mapping is also being used to improve resistance in African crop. For example, in cassava, scientists have mapped resistant genes against cassava mosaic disease, and in banana, against banana xanthomonas wilt. These diseases are major threat to food security in developing region.
And in rice, QTL like MQTL 2.5, MQTL 8.1, and MQTL 9.1 have been linked to blast resistance. That's the fungal disease caused by Macnaport oryzae. These discoveries are crucial because rice is a staple food for more than half of the world's population.
What's amazing is that all this genetic information is now being used in MAS. With MAS, breeders can select plants carrying and desired resistant genes early in the process, saving time and reducing the cost of developing new varieties.
Exactly, and then there's genomic selection. Unlike MAS, which focuses on a few genes, GS, however, uses whole genome data to predict a plant's performance. This is especially useful for traits controlled by many genes, like yield stability or multi-stress tolerance.
Genomic selection also teams up well with machine learning and high-throughput phenotyping. That means we can scan thousands of plants with sensors and drones, feed the data into a model, and predict which one will perform best under climate stress.
So, to sum up, genomics isn't just a research buzzword. It's delivering real measurable results. From durian food quality to rice yield, maize stress tolerance, and disease resistance in staple crops. the benefits are clear.
Plus, it improves food security, supports climate-resilient farming, reduces agrochemical dependency, and creates crops that are more nutritious, sustainable, and economically valuable, especially in vulnerable regions.
And with continued investment in infrastructure, training, and technology, these benefits can reach smallholder farmers across the globe and not just for industrial farms.
Exactly, that's what we aim for. Future where science and sustainability works together to feed the world efficiently and responsibly. Alright, we cover how genomics help identify and improve key traits like yield, stress tolerance, and disease resistance. But genetic is not only part of the story. Let's shift focus to the next omics layer, proteomics, where we look at what's actually happening inside the plants when it's under stress.
Exactly. While genomics tells us about the potential, proteomics reveals the actual response. It studies the structure and function of proteins, which are the real workers in the cell, the enzyme, transporters, and signaling molecules responding to the environment.
So, to understand how crops really respond to drought, heat, or pests, we need to study which proteins are produced, how much, and when. It gives us a live snapshot of how plants cope with stress at the cellular level.
Let's start with drought. Proteomic studies in rice have identified increased level of dehydrins and late LEA proteins. This protein helps to retain cell integrity and to prevent damage from dehydration.
For heat stress, crops like wheat show a spike in heat shock proteins such as HSP70 and HSP90. These proteins help other proteins fold correctly and prevent cellular damage. They act like molecular bodyguards during extreme temperatures.
Also, in salt-stress crops like Brassica napus, proteomics revealed increased activity of antioxidant enzymes, including SOD and APX. These enzymes help plants detoxify harmful reactive oxygen species.
To add, proteomics can identify transcription factors like DREB, WRKY, and MYB, which regulate genes that activate stress responses. These proteins are key in adjusting both abiotic and biotic stress conditions.
Another fascinating part is calcium signaling. Under stress, plants release calcium ion that activate protein like calmodulin, which then trigger the MAPK cascade, a signaling change that boosts plant defense.
Now, moving on to food quality. Proteomics has helped identify proteins that control grain development, aroma, and ripening. In jasmine rice, for example, aldehyde dehydrogenase is responsible for producing 2-acetyl-1-pyrroline, that aroma compound.
In Musang King durian, proteomic analysis revealed proteins involved in sugar metabolism, ripening, and sulfur compound biosynthesis, all of which influence the fruit's distinctive taste, texture, and aroma. This kind of insight is extremely useful for breeders targeting premium food quality.
Proteomics also plays a huge role in food safety. For instance, it helps to detect allergenic proteins like alpha amylase inhibitors and trypsin inhibitors in cereals and legumes. These will allow for proper allergenic labeling to reduce the risk of consumers with food allergies.
On top of that, proteomic techniques are being used in post-harvest monitoring to identify pathogen-related protein. This helps detect early contamination in food processing, ensure cleaner, safer food reach the market.
And because proteomics can detect even trace protein changes, it's also useful in verifying genetically modified organisms and checking for unwanted protein expression, something that's increasingly important for both regulation and customer confidence.
So, in short, Proteomics does it all. It helps improve stress resilience, boost nutritional and sensory quality, and ensure the safety of the food we eat.
And to add, together with genomics, it also completes the whole picture. Genomic helps to identify the potential, and proteomic tells us if those traits are really being expressed under real-world conditions.
Exactly, by integrating omics tool, especially functional genomic and proteomic, we creating a smarter and more sustainable agriculture future. Ones that prepared for climate change, population growth and food safety demands. Before we wrap up, let's talk about where all this heading, the future of omics in sustainable agriculture.
Right now, sustainability is one of the biggest goals in farming. Climate change, water scarcity, and soil degradation are pushing agriculture to its limits. Studies shows that if nothing changes, crop yields could drop by 10 to 25 by 2050, especially in tropical and subtropical areas.
But functional genomics and proteomics offer real solutions. Functional genomics helps us identify genes responsible for stress tolerance, nutrient use, and yield, like OSB ZIP23 and OSP IP21 in rice, which regulate drought responses.
Exactly. So these genes are used in marker-assisted and genomic selection to create crops that can grow in tough conditions with less fertilizer and pesticide use. That means fewer chemicals, lower costs, and more sustainable food systems.
And proteomics add another layer by identify the actual stress responsive protein like dehydrin, LEA protein, heat shock protein 17 and antioxidant enzyme. Breeder can track how crop perform under heat or drought and select the best performing line.
Plus, these proteins act as biomarkers, making it easier to develop climate-resilient cultivars. And since these crops are naturally more resistant, there's less need for chemical treatments. That's a win for both farmers and the environment.
Looking forward, the integration of multi-omics approaches, combining genomics, proteomics, transcriptomics, and metabolomics, will give us even deeper insights into crop biology.
And to that, AI, machine learning, and high-throughput phenotyping technology will be able to predict trait performance faster, optimize breeding, and respond to climate threats in real time.
So the future is clear. Omics tools are reshaping agriculture to be more efficient, more resilient, and more sustainable. With smart science and smart farming, we really can feed the world responsibly.
Okay, and I guess that's a wrap for our topic today. Thank you for joining in. And with that, we'll wrap up. So stay curious, stay grounded, and eat smart. Bye.
Bye.